So we've talked about analyses and measuring chemicals in the environment. So what do we really need to know to do a risk assessment? We talked about risks and benefits. One key thing to remember is the difference between a risk and a hazard. So we're going to start out today. I want to take a quick lead today. So what's the difference between risk and hazard? Any ideas? No? Okay. Professor 1, we'll start out. So, the difference between risk and hazard is if I had the most toxic chemical known to humans in this bottle, would it be a risk or a hazard?
Risk.
Well, if I also told you it's impossible to open this bottle, can't be done. So the material that's in there is still hazardous, right? But if there's no exposure, there's no risk. So what we're going to talk about today is really the toxicological side, what the effects are. So for chemicals to cause problems, they have to be an exposure. If there's no exposure, it doesn't matter how hazardous it is. And there has to be an effect. So if there's an exposure, but it's innocuous, it doesn't cause toxic effects at that concentrations to which the animals are exposed, then there's no risk. Now, the other difference between risk and hazard is risk always represents a probability or a chance. Okay, so this is, if it's hazardous, it's either hazardous or not hazardous. A risk might be, if I told you the risk of drinking this water for you is 1 in 100 million. Would you still drink the water? Yeah, probably. The risk is low. If I told you the risk is 1 in 2, now what do you think? Not so good, right? You're not interested in doing that. So the probability is risk. What is the probability of something happening? Not really the magnitude of something happening. Now the magnitude of something happening is referred to as the toxic potency. So how potent is it? And the effects that occur have the term severity. So if I told you if you drink this water and you won't feel good for the morning, but then you'll be fine, that's pretty low severity. But if I told you you'd be dead by 1 o'clock, that's pretty severe, right? So we talk about the end points. What are the end points? So they can range from minor molecular changes that are transient, that don't last long, or they can be longer term, more severe. We have differences between ecological risk assessment and human risk assessment. For humans, we're concerned about chronic effects. We're interested in maybe not very severe effects, but might have health effects. But for wildlife, we really have only major endpoints that we're concerned with. So today I'll talk about the mechanisms of toxicity which apply to humans and wildlife, and then I'll do an ecological risk assessment. So the ecological risk assessment, the endpoints are what we call apical endpoints. So like apex is the major. Another term for it is canonical. The Bible is the canon, right? So it's the most important. It's not everything, but it's the most important. So wildlife, what do you think the apical endpoints for a risk assessment are? So what would happen to wildlife that we might be concerned about? And I'll tell you, I'll give you a hint, there's 3. What do you think? Any ideas? Well, what would you be concerned about to protect animals in the environment? Any ideas?
Population.
Population. So population is an aggregation of individuals. What determines a population?
Size.
in effects on individuals. So if individuals die, then the population can go down, right? So one of the apical endpoints is lethality. And I'll be telling you some other terminologies that are different as well. So lethality means it will kill it. Mortality is a rate. So the mortality rate is how many die out of a population. So one endpoint is lethality. What's another one? But you're on the right track. It's all about populations for wildlife. It's not about the individuals. For humans, it's about individuals. We care about whether Uncle Louie gets cancer or not, right? But for wildlife, it doesn't matter so much. We're interested in the sustainability of the population. So we've got mortality. What's another one that would affect the population? Because you're absolutely right, it's about populations.
Birth rate.
Pardon?
Birth rate.
Reproduction, right? So that's the second one. So we're going to talk about survival, reproduction, and the last one is growth. Okay, those are really the endpoints that we measure. Now we might measure other endpoints like molecular endpoints to try to predict those, but from an ecological standpoint, we're most interested in those three endpoints. Anything that will contribute to survival, growth, or reproduction. For humans, we've got several endpoints. One of the most critical one is cancer. We'll talk about that today, about the perfluorinated chemicals, okay? Whether they cause cancer or not, and how they might do that. So when we started on this work, there was really nothing known about toxicity, and it was assumed that chemicals like PFAS were inert, they didn't react, and that they were non-toxic. And the reason people thought that was they had, I told you before, they had actually used PFAS as artificial blood. So on the battlefield, what kills people mostly is shock. When someone's injured, then they go into shock, and you want to prevent that. And the best way to do that is with a blood transfusion or plasma. In World War II, a lot of the fighting was in very tropical, hot areas. You couldn't keep blood available. So in response to that, PFAS was developed as an artificial blood for use on the battlefield to save lives. And so because of that, they were pumping this into people, and it didn't seem like it did anything. It was like, well, they survived, actually saved their lives, so that was a good thing. And so because of that, not much was done for toxicological concern. The other thing, remember I told you for the major products that were the perfluorooctane sulfonate-based chemicals and the carboxylate or the PFOA-based chemicals, there was in the United States something called the polymer exclusion principle that said if it's in a polymer, it's not going anywhere, so we don't really worry about exposure, and if there's no exposure, it doesn't matter whether it's toxic or not, and no analytical methods were needed because we didn't need to know if there was any exposure. So it was because of this law, the government regulators like EPA were not asking for any toxicological information. So it just wasn't there. So we had to start from scratch. So the first thing we would like to know is if it's toxic, how does it cause toxicity? And in a lot of our studies, we start with in vitro studies, which don't include animals, cell lines, molecular biology, and try to get an idea of what that mode of action is. So one of the ways to do that is with DNA microarrays, where we would extract some DNA, and then we have the DNA from a number of different endpoints or pathways, generally relating to structural or functional proteins, and then we can hybridize that, and we can then determine what effect occurs. So in something like this is a DNA microarray, and when you hybridize this, if it's green, it means it upregulates that particular gene. If it's red, what does it do? Downregulates. And if it's yellow, it's neutral, nothing happens. So you can look at this picture here and see there are things when we expose the cells, in this case, to PFAS, some things went up, some things went down. Okay, that's great. Now I've told you that. What can you do with it? Not much, right? You really don't know. You don't know what any of these dots represent. But I do. I know what's in each one of these wells. So from that, I can get a gene tree, which will tell us, and you can see up here the way this is set up, it's showing things that are related one to the other. So up here, if things are close together, that means they're really affecting similar things in a pathway. And up here where things divide are different pathways. So one might be respiration, one might be metabolism, one might be some reproductive endpoint. So we have all of these different things, and we can see down here when we expose animals, or cells in this case, to PFAS at different concentrations, different things happen. There are different patterns. But still, you don't know what that means. You can just look at this and go, oh, something's happening. Some genes are going up, some are going down. What is the significance of that? So this is the kind of thing we do. These are pathway analyses, and there are many of these, but the Kyoto pathway analysis is available online. You can get other ones that are very, very similar from the CDC in America. And these are pathways, and they're generally set up to be those that are fairly general. So these pathways are similar between rats and mice and humans. There are a few things, but there's not a lot really to separate us from mice. Mice are mammals, so many of the metabolic pathways are the same. And that's one of the reasons we use them as experimental animals. Not that we care so much what happens to rats, but we want to know what might happen to a human. So it's one model that we use. And what you can see here is if we have PFAS treatment or control, different things are happening. So now when I see something is going up or something is going down, so here's one that's blocked, and I don't expect you to be able to read all the details in here. The point is we can determine which genes are upregulated and which ones are downregulated. So one of the things that we can determine from that then is PFAS alters expression of peroxisomal but not mitochondrial fatty acid metabolism. So the key here is really fatty acid metabolism. If we go back here, we can see in this pathway a gene that would transform fatty acids to that next step is blocked. Now I told you chemicals like PFAS are, remember what I told you? What are they? They're a certain kind of analog fatty acid analog, right? So when they go into the body thinks, oh, here comes a fatty acid. I know what to do with that. I have a pathway. It comes in here. I can transform it. I can use it to make membranes. I can use it to can make hormones, because it can go from there to all these other pathways. But there's sort of some critical steps, and PFAS blocks that. PFAS comes into the pathway, it gets associated with enzymes, but it cannot be metabolized. Remember, because of all these perfluorinated bonds that are very strong. And the other thing about these chemicals is not only do they have fluorines at all of the valence electrons, which are bonds that are basically impossible to break biologically. In addition to that, the way they tend to twist the molecule. So if you can think of a cable or a rope, what do we do with the rope? It's got a lot of strands in it, but what do we do to make it stronger? We twist it, right? And that here means the enzymes, because of that, cannot get to do metabolism. They cannot get to those carbon bonds. So that's why these are forever chemicals. They just don't break down. So there's a problem. So it enters into the pathway, it's cruising along the pathway, and gets to a point where it's binding to an enzyme and it cannot be transformed. So it blocks that pathway. And one of the things that tells us that is that downstream from that, or right where it's being blocked, that gene is overexpressed. Your body says, oh, here it comes, I've got to transform this, but it's not able to do it, so what would the body think? Oh, I'll just make more enzyme. That will solve my problem. And so we see that happen where there's a pathway blocked. We see an upregulation of that gene. So this told us a lot about how these chemicals were working. And because of this, if you block that pathway, you have a problem downstream. You really can't make membranes. You can't make hormones. Hormones are cholesterol-based. So it really has what we call pleiotropic effects. Pleiotropic. It means not just one effect, but a whole cascade of effects. So when this happens in the cell, everything goes haywire. It just goes bad. So there are a lot of other key things that happen downstream, but this is the critical step, or the critical toxicity, and that's what we base our measurement of potency on, because it's the critical mechanism of effect. Okay, so we know something about this. PFAS doesn't include all the genes, but one of the things it does is it does affect peroxisomal proliferation. That can lead to DNA damage. So that's one way that PFAS can cause adverse effects, because it can damage DNA. And if we damage DNA, it can be repaired, but ultimately that's what would lead to stage 1 cancer. So these chemicals do have the potential to cause cancer. So the structure of these is similar to endogenous fatty acids. The physical chemical properties are that they are surface agents. So what can that tell us? Now, I told you the story the other day of the blood bile acids. So what are bile acids? They interact with fat, but in doing that, they're really acting as detergents. Detergents are surface active molecules. So let's see what happens with membranes, because membranes are really critical in the body. A number of enzymes are associated with membranes, and if you affect those, then you affect ion exchange, nerves can't function if sodium and potassium can't move across membranes. And really all other physiological processes would be affected. Metabolism, enzymes associated with that are really all on membranes, whether they're on mitochondria, but they're all membrane related. There are very few enzymes functioning just floating free in the cytosol of the cell. So things are related to membranes. I'm going to talk about one key thing, and that's called, we call it GJAC. But this is related to membranes. And what it means is it's cell-cell communication. Transmission between cells. So in your body, you have blood circulating things, but ultimately biomolecules move between cells. And they can do that a lot of different ways. So let's look at a membrane. This is a pore, a membrane pore. And so in nerves, sodium and potassium move through these openings. And if you block that opening, you inhibit cell-cell communication. Now, a couple other things to notice here are, this is the phospholipid bilayer. So this is a phosphorus head. These are lipids, and that's what makes up membranes. And this is cholesterol. So in membranes, they need to be able to move. So for enzymes to function, they have to really move and rotate. When they're bound onto the membrane, that membrane has to have what we call fluidity. It can't be solid. It can't be locked in. It has to be able to rotate. And one way it does that is it has cholesterol interpolated into the membrane. And that's places where the membrane can flex and bend. Okay? PFAS to the cell looks like cholesterol. So when it comes into the cell, it can enter into the membrane. And so PFAS is really rigid. It's like driving a bar into the membrane. So what do you think that does? It affects membrane fluidity. So some of the things we measure in toxicology are membrane fluidity, which we can do with a flow cytometer, but we also measure membrane charge potential and things like that. But for this discussion, it's really this membrane fluidity that's affected by PFAS. So we know it's going to affect lipid metabolism. We already showed that. It blocks it. And now it's also going to affect membranes. So it enters into that. And this is the GJAC, or the cell-cell communication gap. So this is sort of what that looks like. So we've got our membrane here, and things are moving from cell to cell. So why is that important? Cells work as a team. You have to have communication among the cells. If you inhibit that, the cells basically can't do their job. But more importantly than that, if you initiate cells to become cancer cells by damaging the DNA, then the cells normally will repair the DNA, but if they can't repair it, then the cell becomes rogue, becomes a cancer cell. The cells around it sense that. They say, uh-oh, George is going off the rails. He's not doing things right. We don't want him. And they will kill him. But the only way they can do that is by cell-cell communication. Okay? They can sense that the cell has become cancerous, and instead of it proliferating or metastasizing or metastasizing or growing into a tumor, the cell will kill it. But if we block that communication, then we inhibit the ability to protect by killing the cells. So gap junction intercellular communication, or GJAC, plays an important role of maintaining normal function. It provides, it turns out that PFAS, anything with a carbon chain between 7 and 10, will inhibit GJAC. So one of the things we can do, if we have a whole mixture of PFAS, is if we can determine the critical mechanism of action for all of them, then I can determine relative potency of each one and then add those up in a risk model. And that's going to be critical for people working with PFAS. It's not just PFAS now, that's banned. That's a historical thing. Concentrations are going down. PFOA, historical thing, going down. But there are other chemicals out there. One that's being used as a replacement is called Gen X. We've been studying that. So you're going to have a whole mixture. There are about 1,900 known PFAS in the environment. And we can't do risk assessments on each one individually. Not that would be time-consuming and expensive, but it wouldn't give us the right answer. If you looked at just one, you might be okay. If you looked at 2 or 3, but the effect of all of them together might exceed the critical level. So it's good. We need to know what the relative potencies are. Once we've determined the key or critical mechanism of toxicity, then we can base that relative potency on that enzyme. So let's look at a couple experiments to try to do that. How would we determine the relative potency? We've demonstrated that it happens, and now we want to run rapid bioassays to get the relative potency. So the way we would do that is in vitro, we grow some cells on a Petri plate. We would expose them to our chemical of concern, PFAS, and then we put on a dye. And the dye we use is called Lucifer yellow. We use it because it cannot cross cell membranes. The only way it can get from one cell to the other is through what? The gap junctions, those spaces between cells. And Lucifer yellow is fluorescent. So it's a big molecule, it's fluorescent, it doesn't get metabolized, and so we can use it to determine how much is moving between cells. So we would take our cells, we've treated with PFAS, we put on our dye, and then we scratch across the cells. That cuts them open. Now the dye can go into the cell that we've cut open. Remember, it can't cross membranes, but if we cut the membrane, it can get into the cell. And then if we measure how much goes from that cell into the next cell that we didn't cut, that will be indicative of how much it can cross through these gap junctions. So we scrape that and we wash off the excess dye. We let it sit for a little while, a couple minutes. We wash off the dye. We look at it under a fluorescent microscope and we collect the data. So here's what it looks like. And here are our cells. You can see all the individual cells. Here's where we cut across, and that meant that the Lucifer yellow can get into these cells. And see how bright it is? That means there's a lot of it in there because it's fluorescent. And it moved to this cell, and to this cell, and to this cell, and to this cell, to this cell. And the only way you can do that is through these gap junctions. And you can measure how far it moved in that period of time. And that's an indication of how much you've affected the gap junctions. So here's our control. If we expose them to some PFAS, what happened? Does it move as far? It still gets into the cells that we cut, and it still moves a little bit from one cell to the next, but less. if we get to 50 micromolar of PFAS, it completely inhibited movement. It can only get into the cells that we've scored or damaged. A little bit moving out here. And then by the time we get to 160, it's completely eliminated. So now we can measure this endpoint for a lot of different perfluorinated compounds and determine their relative potency. So we can relate them, and we relate them all to PFAS. We know what PFAS does, and then we can measure what all of our other chemicals do and come up with their relative potencies. And that'll become important later when we do a risk assessment. So that was in vitro, right? You believe it? It's not real, right? It's in vitro, no animals involved.
Can I ask a small question?
No. I'm just kidding. Of course, you can all ask
No,
questions.
I wanted to ask, it's a tissue culture. What kind? It's a human cells or rat cells
Those were liver cells.
from rats?
Yes.
In petri, like a tissue culture.
Yeah, not skin cells, not
Okay, no, I'm just showing like more.
Right, and there are cells that are in culture. There are a certain kind of liver cell. Your liver has many different kinds of cells, so this is just one kind to demonstrate the effect. Now, I don't do rats. I'm allergic to rats. But my friends at the US EPA do a lot of studies with rats. And so what I'll show you now is, because if I go to an agency or a group of people and say, I did this with these cell systems, they're going, yeah, well, but is it real? It's not real, that's just artificial. What does it really happen in animals? So here we do another study. This time we expose the rats and we remove the liver, break up the cells, put them on a plate, put our dye on again, wash it off, go to the microscope, and measure it. So what do you think is going to happen? Do you think it works like in vitro or not? Any ideas? Why don't we vote? Who thinks it's going to prove our hypothesis, or how many think it will disprove? Anybody for proving it? You think? Some of you are asleep and don't care. Just okay, well, let's look and see what happens. So we expose the rats, we take out the liver and we cut it, and then we measure how much it moves. And here's the control, and here's one exposed in the diet to 5 milligram per kilogram body weight per day for 21 days, and we get inhibition. So this was one way we were able to demonstrate that yes, it does happen in animals. Because in the animal, you have all the metabolism going on, you have all the distribution going on, because we're putting it in the diet. So this became one of our critical endpoints, and one that we had bioassays for, that we could screen a lot of chemicals very, very quickly. We use this to determine relative potencies, and one of the things we determined was that the critical structure of the molecule was chain length. Remember, these can be either straight chain or branched, and it's really the number of carbons, and the straight chain turn out to be more toxic than the branched, which is different than a lot of other molecules. Alcohols, for instance, are toxic, but the branched are more toxic because they have a bigger molecular volume per mass. But with these, because of where they're sticking into the membranes, what they're doing and acting like fatty acids, they're more potent when they're linear. And one of the key things that we discovered that affected all the regulations around the world was that the chemicals that were most potent were 8 carbon chains. When they got bigger than that, they weren't as potent. When they got smaller than that, and so now around the world, the regulations are written such that you can use chemicals that are 5 carbons or less. And one of the things we determined was perfluorooctane butanol, PFBS, was much less accumulated and much less potent than was PFAS. And it turned out PFAS is the most useful in industry, and it was also the most toxic. Now, the critical thing about this was when I was doing this research, and when we said PFAS is a problem, and I was advocating that it be banned, it meant we couldn't make these. Okay, can you imagine your life without these? So there was a lot of pushback against me from industries, because it wasn't just the industry making the chemical, it was all the uses that it was put to, but making flat screen devices was critical. Just as a diversion as to why that's the case is what limits the size of our devices is they have microchips in them. And what a microchip is an electric circuit printed on a silicon chip board. And what limits the size of these is how close we can put those circuits together. If we get them too close together, then they will arc across and short out and not work. 2 of the things that affect that are dust. If there's any dust when you're making these and you get that on there, your phone won't work very well because we've made these circuits so close together in here. And the other one is water quality, so ions, and so we have to have ultra clean water. That's why all the chips in the world are made in the United States. They're not made in China, they're not made in Japan, they're not made in Korea, because they can't get the environment clean enough, they can't get the water clean enough. So all the chips are made in one place and then sent everywhere else to make these things. So that's critical. PFAS is a surface active compound, it's ionic, and the key thing about it was it would reduce the surface charge and minimize the amount of dust that would stick onto the chip. So it was critical to make microchips. What we found was that PFBS was much, much less toxic, and it would still be effective in allowing to make microchips. So that was really critical. Once we discovered that, it allowed industry to switch from PFOA and PFOS to PFBA and PFBS. Now they're not as useful, they're not as effective, and they are still persistent. And we now see them increasing in the environment, and they're not without effects, they just cause less effect. The probability of effect is less for them than it is for PFAS. But it's not like they're perfect. And so that helped us sort of move along relative to regulations. The other thing is then, well, one of the things we found was at the critical concentration, the lowest concentration, that PFAS wasn't acting as a stage 1 initiator of carcinogenesis. What it was doing was inhibiting the gap junctions so that if you were initiated, as we all are all the time, every day, that would result in cancer. So you had to read, things like benzoylpyrene are a complete carcinogen. They cause DNA damage at a critical concentration, and they also block cell-cell communication. But PFAS was blocking cell-cell communication at concentrations much, much less than where it was affecting the DNA or causing DNA damage. Okay. Need some water. Oh my God, it's the toxic stuff. I think it's okay. Okay, so I told you I don't do rats and mice, but my colleagues at the US EPA do, and it was this study I just showed you. We were feeding to PFAS to rats and to mice, and we weren't only measuring molecular changes that I was measuring in my laboratory, but we were measuring an apical endpoint. What are the three apical endpoints again? Survival,
Mortality.
reproduction. Okay? When we were feeding this to the rats and to the mice, we get what are called dose-response curves. As you add more and more exposure, the effect goes up and up until it reaches some maximum. And that maximum is called efficacy, how effective it is. When we do risk assessments, as we increase up that line, we can say, okay, where on that line can we be and only have 5% effect? Or where can we be and only have 1%? And so different agencies have different probabilities, but generally for cancer, it's 1 in 1 million. So the risk assessment would say, you know, here we could have maybe 2 cancers out of the population of Belgrade, so not many. But what we saw with the rats was a dose-response curve that for reproduction, and we were focused on reproduction, was no effect, no effect, no effect, and then when we went from this concentration to just slightly more, we had 100% effect. All the babies died. Now this was very scary to risk assessors because it meant if I'm here, no effect, and if I'm slightly over here, there's maximum effect, maximum probability of occurrence, 100%, and maximum severity. They died. Okay? It wasn't a matter of they were sick or didn't grow, they were dead. And this is a problem because how close can you get to that line before you go over it? And the answer we were getting was not much. It was just no effect happening, even though we were feeding PFAS to the dams, to the mothers, to the rats. But as soon as we went slightly above that, we had 100% effect. This was very unusual and very scary for a risk assessor because they don't know how close they can get to that. So we wanted to know, why does that happen? And the critical effect on all of you is it affects the duration and intensity of exposure. So, as an example, if I gave you a big dose of PFAS right now, you wouldn't die immediately, but you might die in a day or two. But if I give you the same total amount of PFAS over a long period of time, and that's how we're all exposed, right? We're not chugging PFAS, we're getting exposed to small amounts over a long period of time. So the question became, why did this effect occur? And my theory was, when it goes into body, it's inactivated by being not metabolized. Remember, it can't be metabolized, but by being bound somehow. So the body protects it from other contaminants this way. As an example, a chemical I've studied a lot, an element, cadmium, you're exposed to small amounts over your lifetime, but the way the body deals with it is it sequesters it. It binds it up. There's an enzyme, there's a protein called metallothionein. And so if you're exposed to small amounts over a long period of time, no problem. It binds it up. So my theory was the same thing happens with PFAS. We know PFAS binds to proteins. And so the question was, in the blood, what protein would it bind to and how tightly would it bind? I'm not going to go into all of the way we solved that, but in general, we used our mass spectrometer, but we had to get the protein and the PFAS through the mass spectrometer, and they both have very different properties. So we had to be able to do that, And it took some doing and some creativity on our part. And one of my assistants, Dr. Paul Jones, was really the key guy working on this. So we mixed our PFAS with proteins and put them into our mass spectrometer, which is shown here. And then you can see where the protein comes out. And in this case, we're looking at bovine serum albumin. So albumin from the blood of cows. And then you can see where the PFAS is. And so the PFAS was binding to the protein. We could show that. We know that albumin has 9 binding sites. Albumin's important in the blood to carry a lot of things around, like hormones. And some are very specific, and some are not as specific, but human albumin has 9 binding sites. And so what we were able to determine was, I'm not going to go through all the details for you here how we did that, but the answer was, yes, it binds to this protein. It binds very, very tightly to that protein, and it binds to only 1 of the 9 binding sites. And what we were able to determine was the strength with which it binds to that site. So we knew the binding capacity, and we knew the binding strength, and from that I can predict how much would be bound and how much would be free. And what that meant was we could predict what would happen in people. So if you're exposed to a small amount for a long period of time, it will be bound up by serum human albumin, and then that ultimately will be processed by the liver and can be excreted. If you're exposed acutely, then bad things happen. And what was happening here was, as long as the dose was small enough and chronic or long-term enough, it was bound up and there was really no effect in the body. But as soon as we got to the point where we saturated the binding sites, then that PFAS was free to do all the bad things it can do, and it killed the babies. Okay? Make sense? Sort of what's going on. So these are the kinds of things that I work on and try to figure out. And you might think, oh, well, if binding the proteins, that's sort of a trivial thing. But it became really, really important in setting the rules and regulations on how much you could be exposed to over different periods of time. So it became critical in all of the regulations that came later. More detail here, you can look at this. I've left all these presentations and every paper I've ever written here, so you can get those. Okay, what are we going to do for break and things? What's your schedule?
You can just tell us when you want a break.
I don't need a break, it's whether they need a break.
Well, I don't know, maybe another 15 or 20 minutes and then we can...
So what I want to do now is sort of change things and talk about ecological risk assessments. So let's just pause a minute. Do you have any questions? Any concerns? I mean, you've got all that PFAS in your blood. So it's a personal thing. I do too. But do you have any questions? Anything questioning, bothering you? Yes, sir.
If it's there is a constant exposure to a small amounts of PFAS, and it's intaken by the body, and it's also bound to the albumin in the blood.
Yeah.
Does it ever get clean? So does this, is it permanently stored, or the blood is getting metabolized and sometimes it gets clean if the organism is not more exposed?
I'll take a little bit of a break while I answer these
Okay.
questions. So that's a good question. His question is, it gets bound to the albumin, and it's not available when it's bound to the albumin, but there's a limit, right? There's a limited capacity to sop it up, to bind it up, and what happens to it ultimately. It turns out that PFAS, unfortunately, one of the problems has a half-life in your body of about 4.5 years. So once, and that it becomes important in the whole assessment on about how much you're exposed to for how long, because you've got what's coming into the body and what's going out. But with that long a half-life, with, you know, caffeine in your body has a half-life of a couple hours. But now you've got PFAS 4.5 years, a lot longer. So things can build up. And it is eliminated. It can be conjugated in the liver and excreted. But there's a problem with PFAS. Now I told you PFAS is a fatty acid analog. And remember the problem we had with the analysis with the blood bile acids. So PFAS to your body looks like bile acid. And your body wants to keep the bile acid. It's hard to make, it's expensive energy, and so once it's done its job as a surfactant, as a detergent to scavenge fats from the blood and take it to the liver where it's metabolized, and then your body will recycle it, okay, and many, many, many times. What that means is for PFAS, there's something called interhepatic circulation. And when your body tries to throw the PFAS out, it says, nope, not going anywhere, I'm going to pull it right back. So it's really difficult for the body to get rid of the PFAS. Okay? That's it in a nutshell, without getting into all the enzymes that are happening. The long and the short of it is it's hard to get rid of it. But you can, a little bit each time. Your body's never 100% efficient in anything it does. So over time, a little bit goes out a little bit, but very slow. Very, very slow. Good question. Good question.
You answered it, so already.
Okay. So anything else, let me know if it's bothering you. Okay, I'm ready to go again. So one of the things that needed to be done was not only understand the toxicology, so we figured all that out, but I'm not a physician, I'm not a medical doctor, I'm a veterinarian. I don't do studies with people, but a lot of other people did. They started doing various epidemiological studies and things with humans. So my job, because these chemicals, remember from yesterday, they're widespread in the environment. And one of the issues was effects on wildlife. Now humans, if we're being exposed to this through McDonald's hamburger wrappers or microwave popcorn or whatever, it's pretty easy to stop that exposure, right? Well, wildlife are exposed, they don't have options to change what they're eating or move out and go somewhere else or clean the dust from their house. They're exposed, and so it was critical to know for regulatory purposes what the effects on wildlife were. So now we had reliable analytical methods. I could measure this compound in the environment. I could measure it in animal tissues and blood and liver and eggs and all kinds of things. And so I could now generate apical endpoint hazard information for wildlife. So the first thing was we knew the target tissues were where key effects happen, and it's these membrane effects, but the liver really is a target, and so we would like to measure what's in liver. We've already worked out a lot of the modes of action. We determined the critical endpoint concentration, and we now had measurements of PFAS in the environment. And so what I wanted to calculate were these hazard quotients. Remember, hazard quotient. So a hazard quotient is a measure of how much they're exposed to divided by the critical concentration. So as an example, if the critical concentration in the diet is 5 parts per million and you're exposed to 10 parts per million, that's bad, right? You exceeded it. And that would mean you have, in that case, a hazard quotient that was greater than 1. If your hazard quotient is less than 1, then we would determine that that particular endpoint would not occur. And that's a simple ratio, hazard quotient. Margins of safety are just the inverse of that. Okay? So if it's less than 1, what you want to know is how much less than 1, how far are you from where you'll have an effect, and that's called our margin of safety. So if the effect level is 1 and you're at 1/10 of a part per million, that would be a margin of safety of 10. And so regulatory agencies, risk assessors, have sort of values they like to see. And the bigger that margin of safety is, the better. And they generally like to see it be around 100 or more. So that would be the probability of an effect happening might be about 1%. So that's sort of how that works. So, what animals could we study? We need to work with animals that are ecologically relevant, but also that we can use in the laboratory. I did a lot of work with chickens. I did a lot of work with turkeys because we can raise them in the laboratory routinely. We can get a lot of eggs, and I can do studies where we inject material into the egg, so that controls how much we know we're getting in. But chickens are not real, right? They're very artificial. So we had a lot of data with chickens, but regulatory agencies said we'd like to know more about other wildlife. So the northern bobwhite quail, we can grow in the laboratory. So this is a relevant bird, it's a wild bird, you can release it to the environment and it will survive. But we could grow it in the laboratory. It's a ground dwelling game bird. It's similar to like a Hungarian partridge, which I think you have here.
It's prepelica. Also, non si mangia.
He's translating what
Yeah.
species that
In Serbia, it would be prepelica, small chicken with small eggs.
Right, they have, in Japan and China, they like to eat the egg.
Yeah.
Tiny little eggs with spots on them.
Yeah.
Okay, so here's our animal. So what I need is, I need to have the effect concentration in different matrices. Because depending on what I've measured in the environment, I need to calculate this hazard ratio. So I might know what's in the water. I might know what's in their food. I might know what's in their egg. I might know what's in their blood. I might know what's in their liver. It depends on what has gotten measured. So I need to have my endpoint expressed by all those things. To work with is the mallard. It's just a different animal. We wanted to work with several animals. Again, it's a wild animal, but we can raise it in the laboratory. So here are the results for the quail. Now, there's a lot on this slide. Let's look at all these things. Well, first of all, I looked at effects on males and females. They're both important. And we also measured what effect was happening with concentrations in the egg. We measured a no effect level and a low effect level. So you can see these numbers are different for the females than for the males. I didn't have everything for everybody, but we generally, so the no effect levels were pretty low. These are exposed for 21 weeks. And then these are the different exposures. So this is what we fed in the food. Here's what we measured on a per body weight basis, the allowable daily intake, the ADI. This was concentrations in the blood and concentrations in the liver. So we had that for the quail, we have it for the ducks. So I've got a number, I've got a no effect level. That means that if I'm at that concentration for that measurement, if I was at 10 in the diet, I would have a hazard quotient of 1. I would be right at the threshold for effect. So what we do with this information is we have to derive what are called toxicant reference values. So I'll do this for birds. We've done studies with mink. I had a mink colony, so I could expose mink. And one of the key things is there are uncertainties, right? And so the way risk assessors deal with uncertainty is by applying an uncertainty factor. So if I determine for one species, the issue is, well, what about other species? And so we actually have application factors or ratios that will allow us to predict among species. And then so we would divide our no effect level by that application factor. Some regulators just use 10 or 100. I chose to actually derive values. It's pretty complicated, a lot of mathematics to do it, but we developed appropriate application factors. So they're sometimes called safety factors, because we're trying to protect all birds, not just quail or mink. So we had measurements. So these are in bird eggs, and we had measurements in all these different species. These were in Michigan and the United States, Antarctica, Canada, Sweden, Faroe Islands, Poland, no, Serbia, sorry, Germany, Italy. So these were the measurements in the egg. So what would we call that? That's the exposure, right? We could measure what was in their diet. That's harder, right? You don't know everything they're eating, but we do that. We do a lot of studies where we determine the diet of birds. We could measure what's in their blood. We could measure what's in their liver. What's the problem with that? You have to kill the animal.
Yeah, of course.
To get it, right? Step 1, remove liver. Hard on the animals. So we didn't want to do that. But eggs, by permit, I have to get permits from the government. I can take an egg and analyze that. And it's more proximal to our key endpoint, which is going to be reproduction. Reproduction is generally more sensitive than lethality. And it's important. If you can't reproduce in a population, ultimately it goes extinct. So here's the data for the bird eggs. Here was the maximum, the minimum, the geometric mean. Concentrations of things in the environment are generally not normally distributed. That means we wouldn't compare to a mean. The trends are over-dispersed, they're skewed positively. That means the mean would underrepresent the value, so we use the geometric mean. If you know how to do that, great. If you don't, I can tell you later. But we would use geometric means. And then here's our margin of safety. And then over here is our threshold. This is the value that we chose. Our toxic reference value, or TRV, was 1.7 milligrams per milliliter in water. So what I did was I had to predict, because people measure what's in water, and I had to predict from the water how much would get into the bird and how much would get into the egg. I'm not going to go through all those calculations, but in the papers that I gave you, it's all there. Okay? So ultimately here, what we wanted to do was relate that exposure back to what's in water. Because that's what most people were measuring, and that's what regulations around the world were based on. So I've done that. And so help me interpret this. What do you think this frequency distribution tells us? It's a histogram. Any ideas? How would you interpret this?
How many eggs were how many eggs were identified with that much PFAS, maybe?
Well, actually, how many water concentrations that would relate to how much was in the egg would be there. Exactly. So is it a problem or not a problem?
It is. Well,
These are all less than that value. So these would all have a hazard quotient less than 1. What about these? Over 1. So the conclusion was a lot of birds weren't at a level where the water concentrations that were being measured around the world weren't sufficient that we would be having effects, but some were. Okay, so it wasn't a matter of no effects, and there was no margin of safety. Okay, so when a risk assessor would look at this, they would go, oh, this is a problem. We need to reduce the concentration in water so that we have less in the eggs that would be exceeding this threshold for effect. So it was a problem. And this surprised a lot of risk assessors around the world. And part of my job working with the US EPA was to travel around and talk to governments. I didn't come to Serbia, but I went to a lot of countries and presented information, talked to them, and got them on board because ultimately we wanted to have it banned through the Stockholm Convention. So we can also measure bird livers. And here's a whole bunch of birds now. We have more. And again, I can relate by using a biomagnification factor back to water. So what do you think happened? Did we exceeded the threshold or no? So here we are. So again, most were, this is now measurements actually in the liver, not in water. And so this is more proximal to the effect. And again, most of the birds were below this threshold, which includes a safety factor, but some were above. So this is the kind of information that a risk assessor looked at and said, okay, it's not that terrible, but we're right there. And remember about the dose response curve thing, where when you're right at the threshold, no effect, and then boom, you're above it, everybody's dead. So when they were looking at this, they were saying, this is not good, this is bad. We're in a bad situation, we need, and when I was presenting this information, the decision jointly between 3M company that made these chemicals and the US EPA was to stop production almost immediately. Some companies hated me because when the decision was made, it was made within hours of my data being presented and gave them little time to react or change to other things. So it was very, very precipitous. Okay, we can also measure blood serum. This is why I wanted the endpoint reported, or the exposure for these endpoints reported on all these different things. Remember, I was taking blood samples. Somebody was asking me how I do that. And we do that for a lot of species. And the reason we do that is it doesn't kill the bird, right? I take a blood sample and measure a number of parameters and release the bird. But again, same kind of effect. We were mostly below the threshold at the time, but some were above. So it was a scary situation. Here's some more water values around. It's pretty much all the same thing. So even with safety factors applied in some locations, and we know what these locations are. And so that meant we could advise governments, you have a problem here, you have a problem here, address that first. And I'll show you how we did that. So that's sort of the risk assessment. The risk assessment concluded that for wildlife, and I didn't show you all the other things, we did things with fish, we did things with mink, and the answer was the same really for all of them. That it wasn't terrible, but we were exceeding the hazard quotient of 1, even with safety factors applied. So the decision was that it should be phased out. In addition to that, then, I had a big project by the US EPA for all of the Great Lakes in North America. And what we were doing was measuring concentrations in sediment cores. And you can see here, this is just for one lake. And I had samples from these different places in the lake. And then I could look into the core and look back in time. And see where things really started, and we had this sort of peak up here, and then for this, things are going back down. This is 10 PFCs added together just for trends. One of the things we learned from this was that the PFOA, the PFBS, the substitute that allowed us to make cell phones and computers was increasing in the sediment over time. We see it coming up. It's less toxic, less accumulated, but it's still not non-toxic. And so we presented this to the government and said, because I told them this would happen. And then they went, yeah, sure, right. But we've now demonstrated that it is happening.
Sorry?
Yes.
Something? So, do I understand these drugs correctly? So you found PFAS in sediments from 1920,
Yeah.
but were they produced then? Wasn't step 1 produced in...
Yeah, no, that's a good question. This is a total of 10, and the solid lines are the extractable organic fluorine. So it's not just the synthetic compounds. There are 1 and 2 fluorine molecules made naturally, and they would be included in the extractable organic fluorine. But good question.
It's like a baseline and then...
Sort of a baseline. And it comes from a lot of different processes. But that part of this would be that. What we're really kind of looking at, and I don't have all those details here, it's in the manuscript. But each one of these, and so this one, this is PFAS. It's making up most of it. And we reached the maximum here, and I think this was for, trend was going down in total extractable fluorine. But very good catch, good question. So, a few local places. I've had a number of people in the laboratory from Korea. I do a lot of work in Korea, and this is an area on the west coast. It's called Lake Shiwa. It's an artificial lake. They've built a dam across here. There's a lot of industry here in Ansan and Incheon City. It's on the west coast of Korea. And here are some of the concentrations. This is just PFAS. And so we found very, very, very high concentrations in the sediment in Korea. We presented this information to the government. They went first of all, they controlled the releases. They said stop releasing it. It was from a paper and textile manufacturing industries. So they stopped the inputs, and then they went in and remediated. They dug up all of the sediment and then incinerated that. So it was very, very expensive. But it's a pretty small area. And the result of that was it wasn't 100% clean, but it was much, much better than what it was before. So these kinds of studies we've been doing, we try to relate to regulations like the Stockholm Convention and regulations in individual countries. And we also try to affect quality of the environment and protect people and wildlife. That's what we try to do. So, what is the current situation? I would say the catastrophe was avoided. Remember, we only exceeded the threshold for effects in those species I showed you by a little bit. And that was pretty constant with mammals and water and sediment, a lot of things we looked at, but not by much. Okay? There were probably some effects occurring, there was probably some reproductive effects, but not enough. There are now replacements, so we can still have computers and things, but those replacements are not without risks. The ultimate answer is we need to have chemicals that will degrade in the environment and not persist. I've invented some of those. What I've invented and are patented are things called segregated halo ethers. That means there's an ether linkage, which is an oxygen, between some of the carbons, and that separates the fluorines enough so that chemical reactions can occur. And so these compounds work pretty well, but in the environment they unwind and fall apart. The problem with that is they form trifluoroacetic acid ultimately, which is now come under regulatory scrutiny. Regulators are afraid of TFA, so my chemicals aren't being used, so we don't have that safety factor. And it's true, you know, but TFA is not particularly toxic. But regulators are worried about it, and so they're delaying switching because of that. So concentrations in general of all these things, of the historical ones are decreasing, but the new ones are increasing. They're coming up, and we're finding them. I find them in the environment. So you know, PFAS was based on, placed on the Stockholm Convention, also PFOA. Now 8 chemicals are banned. In my country, in Canada, we banned 87 chemicals, and we banned them back in 2006. So we were among the first to do that. They're still made in China. I've been working in China a lot. You'll see in the papers we've published a lot. And there is a lot of political will of the public to phase these out. But the government still allows them to be made. This is sort of a statement, because I was advising the Minister of the Environment, but on January 1st, 2006, in Canada, we banned all these chemicals. Just more about that. One of the things that I've done is under the Canadian Environmental Protection Act, I wrote all of the policies and procedures on how we handle these chemicals in Canada. And I also chaired the Environmental Review Board for Canada for a number of years. But now I'm old and retired. And that would mean issues like this would be brought to that board. On my list, you'll see there's a paper, it's not on the perfluorinated list, about the Canadian Science Court. And what we've done in Canada is take these very technical issues that we've been talking about for the last few days and put the decision-making in the hands of scientists like me. as opposed to the courts. In America, I'm not sure how things work in Serbia, but in America, when there are these controversies between industry or advocacy groups of the public, the decisions are made in civil courts by people just like you off the street, and you know a lot more than what the average person would know. And they just make these decisions in courts, which is not a very good way to do it when it's a really, really hard, difficult, technical issue. There were a lot of papers, and you can read these, there were people writing a lot of really not very nice things about me, that I was trying to destroy the world's economy and things like that, which wasn't the case. But there was a lot in the public domain discussing me. Some people thought I was a hero and liked me. Some people hated me, thought I was the devil. And there were lots and lots of articles written for and against me. So with that, I'll stop. That's what I want to tell you about. But I want to leave enough time to answer any of your questions that you have. There's a lot more to the story than what I can tell you today. I tried to keep it simple. I tried to give you what you need to make informed decisions or understand how these assessments are done. So with that, if you've got any questions, I'll be glad to answer them. Here's how you can contact me. If you've got other questions or want something, need something, feel free to write. So with that, I'm going to take a rest.
Thank you. Does anyone have any questions?
Questions, comments, anything? First was here.
So you mentioned the epidemiological studies on humans. If I'm not wrong, we have some reproductive problems as well. For example, there is that story about the sperm count falling over the decades, and also I'm not sure how correct that information is. I believe also lots of women have reproductive problems. Are there any links established between PFAS and those problems?
So his question is really epidemiological studies and what evidence do we have that PFAS might be having effects on people like us. Epidemiological studies are difficult because they're not what we call case control studies. So with occupational exposures, we could compare all the people that work and are exposed to a chemical or a group of chemicals and compare them to people who are not. That's called a case control study. Epidemiological studies, where we look at the whole population and we would measure concentrations, and in America and in a lot of other countries, there's a lot of monitoring going on with people. In the United States, there's the Centers for Disease Control every year make measurements of a lot of chemicals, including phthalates and other things, but in human blood and in human urine, so there's a lot of measurements of exposure. And then what people do in epidemiological studies is compare those exposures to effects. The problem is you're exposed to lots of other things. And you can go to the literature and find papers that say, oh, there's an association between PFAS and some adverse effect. But you can also find papers that say there are associations with PCBs and the same effects. So it's really difficult to pull apart what's the causative relationship. I'll show you a paper here somewhere. I think it's here. Sorry. let's see. I'm not seeing it here. Anyway, I have a paper that we've just finished that we related exposure to a number of perfluorinated compounds as causative agents for diabetes. But in addition to the epidemiological studies, we did mechanism studies. So we have plausible mechanisms that show on a molecular basis how it happens. Okay? And that's really the link you need. If you just go out and do a study and say, I've measured cadmium in everybody and then I've determined what the health status of everyone is, it's difficult. But if you can link it and say, well, here's the way it could happen, that's more important. In Brno, at Recitox, where I work a lot, we have very, very large registries of disease for many places in Europe, Central Europe in particular. So we do these kinds of epidemiological studies, but they are difficult. And in the end, you can never prove causality. That's the problem. You can see associations. And we calculate risk quotients, and we try to determine, but it's difficult. Okay? You cannot prove what the cause was because of all these exposures. It's a good question. I wish I had a better answer. For some chemicals, we do have better epidemiological data where, for dioxins, for instance, where we had industrial accidents. So we had whole cities that were exposed, and we could get a spectrum of exposures, and we had all the reproductive outcomes. And it was pretty obvious that there were effects on reproduction in that town in Italy where the explosion happened. But for other places, it's harder. Yeah, it's a good question, and I wish we had a good answer, but
I guess another problem is that PFAS are present everywhere now, so it's harder to compare.
yeah, no, exactly. What he said was one of the problems is it's everywhere, and that's true. There are differences in concentrations. We did a study, an association study with pregnancy outcomes in Korea. And we were able to show associations, but can't prove that that was the cause. Different places have different profiles of exposure. Some are higher, some are lower. But yeah, you're right, they are everywhere. And changing all the time. It's sometimes hard, like with the reproductive outcome and stuff, you need to kind of look back and see